Classification of family Streptococcaceae. Streptococci, on the basis of oxygen requirement, is divided into obligate anaerobes, example Peptostreptococci, and aerobes and facultative anaerobes. On the basis of oxygen requirement, Streptococci is divided into aerobes and facultative anaerobes and obligate anaerobe. Peptostreptococci is an example of obligate anaerobe. On the basis of hemolysis on 5% sheep blood agar, the aerobe and facultative anaerobes are classified into alpha hemolysis, beta hemolysis, and gamma hemolysis. Example of alpha hemolysis: viridans, streptococci, and pneumococcus. Alpha hemolysis means partial hemolysis. Partial hemolysis. So they will create a small zone of greenish discoloration. Beta hemolysis means complete hemolysis, and they will create a large zone. Example: Streptococcus pyogenes and other beta hemolytic streptococci. Gamma hemolysis means no hemolysis. Okay, so gamma hemolysis, it's actually a misnomer because there is no hemolysis. An example is enterococcus. So aerobes and facultative anaerobes, on the basis of hemolysis on 5% sheep blood agar, they are classified into alpha, beta, and gamma hemolysis. Alpha hemolysis means partial hemolysis. Example: viridans, streptococci, and pneumococcus. Example of beta hemolysis: streptococcus pyogenes and other beta hemolytic streptococci. Example of gamma hemolysis: enterococcus. Then we have Lancefield grouping. Lancefield grouping was given by Rebecca Lancefield in 1993. Okay. In Lancefield grouping, we divide the beta hemolysis into 20 serogroups. That is group A to V except I and J. And this Lancefield grouping is on the basis of C carbohydrate antigen. These C carbohydrate antigen are present in the cell wall. So Lancefield grouping was given by Rebecca Lancefield in 1993. And under this, the beta hemolysis, they are further grouped into 20 serogroups A to V except I and J. And this is based on group specific C carbohydrate antigen. See, Lancefield grouping. The beta hemolytic streptococci were further classified by Rebecca Lancefield in 1993 on the basis of C carbohydrate antigen present in the cell wall into 20 serological groups named A to V except I and J. And most of the streptococci which cause human infection, they belong to the group A. Like Streptococcus pyogenes belong to group A. Then it was further classified. Griffith typing is on the basis of M protein, and Griffith typing classifies these group A into more than 150 serotypes. Griffith typing is based on M protein, and this Griffith typing will classify these group A into more than 150 serotypes. Genotyping is based on M gene. This M gene codes for M protein, and genotyping will classify this group A into more than 200 M genotypes. Okay. Griffith typing is based on M protein, and Griffith typing will classify group A into more than 150 serotypes, and the genotyping, which is based on a gene, M gene, it will classify group A into more than 200 M genotypes. So this is the classification of family Streptococcaceae. On the basis of oxygen requirement, classified into aerobes and facultative anaerobes and obligate anaerobes. Example of obligate anaerobes: Peptostreptococci. Aerobes and facultative anaerobes on the basis of hemolysis classified into alpha, beta, and gamma hemolysis. Example of alpha hemolysis: Viridin streptococci and pneumococcus. Beta hemolysis example: Streptococcus pyogenes and other beta hemolytic streptococci. Gamma hemolysis example: Enterococcus. Now, this beta hemolytic streptococci are further classified on the basis of group specific C carbohydrate antigen, and this is known as Lancefield grouping. Lancefield grouping was given by Rebecca Lancefield in 1993. It is based on group specific C carbohydrate antigen. In this, the beta hemolytic streptococci are classified into 20 serogroups from group A to V, except I and J. So most of the streptococcus that are infective to human, they come under group A, like Streptococcus pyogenes. That further classified in Griffin typing. They are classified into more than 150 serotypes based on M protein, and in genotyping they are classified into more than 200 genotypes based on M gene.